Today we will see the chapter 6. Chapter 6 title is Pedigree Analysis, Application and Genetic Testing. So we will see some pedigree and also we'll apply the pedigree analysis, and also we will see some genetic counseling. In this slide we will see some introduction. So everybody has the fingerprinting, everybody has unique fingerprinting, but there is some people who don't have the absence of fingerprinting. These people we call it as odomatoglyphia. So this is the pedigree of Swiss family with odomatoglyphia. So for example, the colored scale or the circle are people with odomatoglyphia. So we will see some details in the later slide. Okay. So in this slide we will see some study of genetics in human is constrained by special feature of human biology and culture. So human has some special feature. First one is that controlled mating is not possible. For example, this is common characteristics of a model organism because for example, people use zebrafish to genetic analysis, while people use fruit flies for their genetic analysis. But in these cases, the controlled genetic crosses is possible. But human, the controlled mating is not possible. And also, human has the long generation time. For example, fruit fly, their generation time is 10 or 12 days, but humans' generation time is around 22 or 33 years. And also, human has a small family size. For example, the common characteristics of a model organism is the production of numerous progeny. But human can produce one baby and most the twins. But for example, zebrafish, at one time, they can make the hundreds of the babies. So this is different. This is some special feature of human biology and culture. So pedigree means that the pictorial representation of human family history. And the family tree like this outlines the inheritance of one or more characteristics. For example, unaffected person, males, we just designate the square, and female, we just designate the circle. And also sex unknown or unspecified, at that time we designate the diamond. And also person affected with the trait, we add color, red color, and we also add red color, red color. And also obligate carrier means that in Korean we call it as a 보인자. The carries the gene but does not have the trait. At that time we just add the dot inside the circle, we just add a dot like this. And also deceased person means that person already died. So in this case, we just add a slash, slash, slash. So proband means that the first affected family member coming to attention of geneticists. In these cases, or we can say that the proband means that the person from whom the pedigree is initiated. In this case, it's just we add P, 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 and arrowhead. The arrowhead, arrow like this. So this is the very basic example. And for example, this is the parent, and this is three children. Parent has 3 children, and the first child was a boy, and second child was a girl, and third child was girl. This is one example. In twins cases, we just add the bracket like this. This is identical, and this is non-identical. And consanguinity means that 건친혼, mating between the related person. So in these cases, we just this one is indicates the consanguinity. Okay. This is the one example of pedigree. So this is pedigree of a person with Waardenburg syndrome. Waardenburg syndrome is characterized by the deafness, they cannot hear, and pale skin, and the visual problem, and the white product like this. This is 하얀 앞머리, like this. This is the Waardenburg syndrome. So actually, this person was proband. This person was the proband means that whom this degree is initiated. So, for example, the proband means that first affected family member coming to attention of a geneticist. So, geneticists find that this person is affected after geneticist and initiated pedigree. So he investigated their father, mother, and their grandfather and great-grandfather like this. So this is the P means that this is the proband, okay? And each generation in pedigree is identified by Roman number, Roman number 1, 2, 3, 4, like this. And this means that father, parents, grandparents, great-grandparents. Great-grandfather is died, but great-grandmother is still alive. So, and also, this means that great-grandmother is affected. And also, field symbol represent family member with a Waardenburg syndrome. And this means that represent unaffected member, like this. And this is the twins, non-identical twins, like this. So children in each family are listed from left to right in birth order, like this. Okay? So this is one example of pedigree. So this is pedigree of a person with the Waardenburg syndrome. In this slide, we will see second subject. Geneticists often use pedigree to study the inheritance of characteristics in humans. First, we will see the autosomal recessive trait. Gene is located in the autosome. So human has 22 pairs of autosome and 1 pair of sex chromosome. Male has XY and female has XX. The autosomal recessive means that gene is located in the autosome. And also recessive trait means that, for example, large R is dominant, small r is recessive. So this is recessive autosomal, but if this is located in the autosome, it means that this is autosomal recessive trait. And also, for example, in this cases, the father was carrier, so father's genotype will be the large R, small r, and mother is also carrier. Mother's genotype will be the large R, small r. So after mating, they have the large R, and also they will have the small r, large R, and they will also have the large R, small r. Final one is large R, small r, small r. So in this case, it's only the children with the disease So this is normal, large R is normal, and this is the also because they have the large R is dominant than the small r. This is also normal. This child is also normal, but this is the child is the children with the disease is the this is the this child has this disease. So in this case, children with the disease will be the 1/4, will be the 25%. Okay, so usually the autosomal recessive trait appears with equal frequency in both sexes because this gene is located in the autosome, and because this is a recessive trait, this is skipped generation. Usually the skipped generation. In first generation, they don't have any symptom, but second generation, one people is affected, and second generation is there is no people who is affected. In first generation, there was a consanguinity, so continuum. At that, the first generation they have some disease. So usually the autosomal recessive traits are more likely to appear among progeny of related parents. And also they usually skip the generation and appears with equal frequency in both sexes because this gene is not located in sex chromosome. So this is the summarized of the autosomal recessive trait. So usually the appears in both sexes with equal frequency and tend to skip generation. So when both parents are heterozygous, approximately 1/4 of the offspring will be affected. So one out of 4 is affected, and also appears more frequently among the children of consanguineous marriage. Okay. In this slide, we will see the autosomal dominant trait. Because this gene is also located in the autosome, not sex chromosome. They are located in the autosome. So because they are located in the autosome, usually they appears with equal frequency in both sexes. And because they are the dominant trait, so usually they do not skip the generation. This is one example. Father, he don't have without the trait, so the father's genotype is small d, and mother was affected. So because this is dominant, so this is she was affected. So she will be the large d, small d, like this is one example. So after crosses, this is after they crosses, the genotype will be like this, and this child will have the disease or will have the trait because this is dominant. How about this one? This child also will have the disease or the traits. So this, and how about this one? This child and this child, they will not have the disease. And in these cases, 50% of children will have this disease, and also 50% of children will do not have the disease. So, okay. So usually the autosomal dominant trait appears equally in males and females. For example, in first generation, father and mother, but mother was affected person. After they crosses, first children was the daughter, second children was daughter, and third children was a male, and fourth children was male. So in these cases, you can see that among two daughters, one daughter was affected. Among two sons, one son was affected. So usually they appears equal frequency 50% and 50% in both sexes. And also first generation, they have a person who was affected, and second generation, they also have the person who was affected, and third generation, they also have the person who was affected, and fourth generation, they also have person who was affected. So in these cases, autosomal dominant traits do not skip the generation. This is difference between the autosomal recessive trait. Usually they skip the generation, but in these cases, they usually do not skip the generation. So in this slide, we will see some example of autosomal dominant trait. One example is familial hypercholesterolemia. The person who has this disease, usually in this is blood vessel, usually because they have some deposit of lipid deposit here and there. So people who have this disease, they have the higher blood pressure, and also sometimes they experience a heart disease or they have some stroke. Usually the LDL, low density lipoprotein, is accepted by LDL receptor. After that, they can move into the cell. After that, the LDL is breakdown. But if there is this patient, they have some defects in the LDL receptor, so LDL particle cannot move the cell. So usually this particle is deposited in the blood vessel. So it induces the higher blood pressure, heart disease, and stroke like this. So this is autosomal dominant trait. The characteristics, once again, usually appears in both sexes with the equal frequency. You see here, the 50% of daughter is affected and 50% of son is affected. And also, do not skip the generation. Every generation, they have the people who has disease. So affected offspring must have an affected parents, like this. Affected offspring must have the affected parents. Affected children must have affected parents. Affected children must have affected parents, like this. So this is autosomal dominant trait. Once again, this is the summarized. Affected offspring must have an affected parent. Okay, so this is a very important trait in autosomal dominant trait. So in this slide, we will see X-linked recessive traits. So it means that this gene is located in the X chromosome, not the autosome. Autosome means that chromosome from chromosome 1 to chromosome 22, but this gene is located in X chromosome. And also they show the recessive trait. For example, the mother, she has large X, and she also large X, and small r means that she is carrier, because small r is recessive, so she is carrier. So she is a carrier mother, and father was normal. After crosses, for example, after post crosses, this X become the normal daughter, and this X and Y will have the normal son. How about in these cases, X, R, and comes from, also they have a large X, so this is daughter, but she is carrier. How about this one? She received the X, R from their mother, and Y from father. So she is. So in these cases, the son is affected. So affected person will be the 1 out of 4. 1 out of 4 like this. So okay. So in this case, because this is the X-linked recessive trait, you will see that appears more often in males than female. So you can see here, male is affected, male is affected, and male is affected. And also, affected sons are usually born from unaffected mother, but the unaffected mother is the carrier. How about in these cases? Affected sons are usually born from unaffected mother. How about these cases? Affected sons usually born from mother, unaffected mother, but she is carrier, like this. So usually both are not passed from father to son. For example, in this case, the father is the affected, but none of their son is affected. How about this case? In this case, is also father is affected, but in this case, none of their son is affected, but their daughter will have the carrier, because she received X small r from the father, and also she received the normal X from their mother, so she is carrier, like this. So this is summarized again, appears more often in the males than the females. Usually the affected son is usually born from the unaffected mother. The mother is always carrier. And also are not passed from father to son. Because if their father even though father is affected, their son is normal. Even the father is affected, their son is normal. So the very famous X-linked recessive trait is hemophilia. So hemophilia is that they have some abnormal factor 8. So factor 8 is very important role in the blood clotting. So if people have hemophilia, so there is no clotting into their blood. So finally, after they have some scar here, usually the blood is coming out and the people will die, eventually die. So this is the hemophilia. So usually the genes encoding factor 8 is located in X chromosome. Okay. So here, for example, in first generation and second generation mother was the normal, but she is the carrier. So usually their son is affected. So in these cases, also Alice is also affected, and also their son is also affected, and their daughter is also Alexandria. King Alexandria is normal phenotype, but she has the affected. She is in But at that, this is a Russian Roman family, a Russian royal family. So usually in first generation, the mother shows the normal phenotype, but she is carrier. So her son Alexis has have the traits like this. So based on this one, so usually the male is more affected than the female, and also the affected son usually comes from unaffected mother, but the mother is a carrier. Affected son comes from unaffected mother, the mother is carrier. Affected son comes from the unaffected mother, but she is a carrier like this. So this is the summarize of the X-linked recessive trait. usually the males, more males than females are affected. And also affected sons are usually born from unaffected mother and never passed from the father to son. So this is the summary. Okay. In this slide we will see another X-linked dominant trait. Previous slide we will see that X-linked recessive trait. This is now we will see dominant trait. Only one copy of disease allele on X chromosome. So the gene is located in X chromosome, but they are dominant. So that's why the only one copy of disease allele on X chromosome is required for an individual to be susceptible for an X dominant disease. So usually this one is affect both male and female. Okay, and also important thing is that affected male usually pass their trait to all of their daughter. For example, the first generation, they have their one son, and also they have 3 daughters. All of their daughter is affected because all of the daughter received the X chromosome from their father. That's why all affected father pass the trait to all of their daughter. So usually they do not skip the generation. We see that in the first generation and second generation and third generation and fourth generation. Okay, in the case of the affected female, passed their trait onto help of their son and help of their daughter. For example, in this case, she only has 3 children, but she has 2 daughters. You can see that the help of their daughter is affected, like this. Okay, and maybe the help of because she only has the 3 children. If she has another the son, maybe the son will be unaffected. The ratio will be like this. So this is the X-linked dominant trait. Both males and females are usually affected, but they do not skip the generation. The most important thing is the affected father pass the traits to all of their daughter. So this is the specific characteristics in the X-linked dominant trait. Okay. In this slide, we will see the Y-linked dominant trait. So the gene is located in the Y chromosome. Okay, not autosome from chromosome 1 to 12-2, but this gene is located in the sex chromosome, especially in the Y chromosome. That's why it appears only in males, and also all male offsprings. For example, all male offsprings of affected males are affected. In this case, all male offsprings of the affected males are affected. In this case, all of the male offsprings of an affected males are affected. In this case, all of the male offsprings of affected males are affected. In this case, it's also same. And also these traits do not skip the generation. For example, in first generation, they have disease. In second generation, they have disease. In third generation, they have disease. And fourth generation, they have disease. So this is the Y-linked dominant trait. How about Y-linked recessive trait? Y-linked recessive trait, we cannot detect the phenotype because the Y is only located in single copy. So if they are recessive, we cannot detect the phenotype. Okay. So the characteristic, this is table 6.1 to summarize the Y-linked dominant traits. Only males are affected, and also all male offsprings or one affected males are affected, and also do not skip generation. In this slide, this is concept check 1. The question was the autosomal recessive traits often appears in pedigree in which there have been consanguineous mating because these traits, so usually this is autosomal recessive trait, usually appears with equal frequency in both sexes and skip generation, and also appears more frequently among the children of the consanguineous marriage 친족 결혼, okay, in which it's because these traits are tended to skip generation. It could be, okay. Third one is that usually arise in children born to parents who are affected. It could be, but appears only equally in male and female. It could be, but if you see here, number 2, number B, appears only when both parents carries a copy of gene for the trait. So the father consanguineous, they are cousin each other, but this is father and this is mother. This father is also carrier, and this, but they do not. He do not show the phenotype. The mother also is a carrier, but she do not provide the phenotype. But usually the appears only when both parents carry a copy of a gene for the trait, which is more likely when the parents are related. So this is very rare cases. That's why autosomal recessive traits are often appears in pedigrees in which there have been consanguineous mating, because the most reliable answer will be the number B. Okay. In this slide, we will solve another question. The question was how could you distinguish between an autosomal recessive trait with a higher penetrance in males? So usually the autosomal recessive trait, they appears with equal frequency in both sexes. But how could you distinguish A and X-linked recessive traits? So usually autosomal recessive trait appears with equal frequency in both sexes, but in these cases they show that higher penetrance in the male means that appear with a higher frequency in the male. And also X-linked recessive traits appear more often in males than female. Okay, how can you distinguish A and with B? The answer will be that this is X-linked recessive trait. The characteristics: affected sons are usually born from unaffected mother. So affected son usually born from an unaffected mother. Affected son usually born from mother, unaffected mother. Affected son usually born from mother, born from unaffected mother, but which has all of them they are carrier like this. So this is the big difference between the autosomal recessive trait with a higher penetrance in male and the X-linked recessive trait. So X-linked recessive traits are only passed to the son from mother, not from father. So this is the answer. X-linked characteristics, X-linked recessive traits are only passed to son from mother to son, mother to son, and mother to son, not from the father. So this is the answer. Okay. So in this study, we will study new subject. Studying twins and adoption can help assess the importance of gene and environment. So this is the monozygotic twins. In twins, there is two subtypes. One is monozygotic, the other is dizygotic. Monozygotic means that they are identical twins. They are identical twins. Their phenotype is exactly the same as each other. But there is also another type, is dizygotic twins. In Korean, we call it as the 일란성 쌍둥이. They are non-identical twins like this. The shape, the appearance is already different. But if we want to study twins, we have to know one concept first. The concept is concordant, the trait. This means that the trait shared by both members of twins pairs. Okay. Next slide. This slide we will see the concordance. Concordance means that the percentage of the twins pair that have the concordance trait. For example, in monozygotic twins, if one twins have the twins is male, they have twins, they are monozygotic. If one twin have the heart attack, and the percentage of the other twins will have the heart attack is 39%. How about in dizygotic twins? If they are male, if the one of the dizygotic twins, if they have the heart attack, and the other twins, the percentage of the other twins will have the heart attack is 26%. Okay, how about epilepsy, 간질? If in between the monozygotic twins, if one twin have the epilepsy, if one twin shows the epilepsy, it means that the percentage of the other twin which will have the chance to the epilepsy is 59%. How about the dizygotic? Dizygotic is that if one twins between the dizygotic twins has the epilepsy, and second question is what's the percentage of the other twin which will have the chance to have the epilepsy? Is 19%. Okay? Is this clear? Okay. So now we learn the concordance, and there is the percentage of twins that have the concordance trait. This slide we can extend the application of a twin study. Now is we can connect with the twin study with the asthma. Asthma in Korean we call it 천식. So, okay, 천식 is caused by the combination of the genetic factor. Because why? Concordance value for monozygotic twins is 65%, and the dizygotic twins is 37%. It means that gene is very important. So, but how about the environmental factor? If one twins, so for example, there is the monozygotic twins. So, okay, one twin live in the area, or for example, which has a lot of dust and pollen and air pollution and respiratory infection and cold air. In these cases, this twin will have the higher chance of the chance of to get the asthma. For example, the other twins, their genetic background is exactly the same, but these twins live in very clean and there is no pollution area. In these cases, even though he or she has the asthma gene, but usually they did not develop asthma very well. So it means that based on twin study and asthma, is the conclusion is the asthma is caused by a combination of genetic and environmental factor. Okay, we can go back to the previous slide. Okay, for example, based on this result, the heart attack, the concordance is monozygotic is 39% and dizygotic is 26%. It means that the genetic factor is very important role in heart attack. How about the epilepsy? There is a big difference between the monozygotic and dizygotic, means that epilepsy is the highly dependent on genetic factor. Okay? So if we compare the epilepsy and heart attack, means that epilepsy is higher dependent on genetic factor than heart attack. Okay? So we can, based on this twin study and the asthma, we conclude that asthma is a combination of genetic and environmental factor. Okay? In this slide, this is concept check 3. The question was that a trait exhibit 100% concordance in both monozygotic and dizygotic twins. What conclusion can you draw about the role of genetic factor in determining difference in the trait? Okay, for example, this is the one example, Jennifer and Karen. They are in the identical twins. They are born with the same gene, but Jennifer usually eat healthy and low-fat food. So Jennifer is very slim. But Karen is that she always eat fatty, unhealthy food. Usually Karen shows the obesity. But in these cases, for example, the weight, body weight, the trait exhibit 100%. It means that their phenotype is exactly same. It means that genetic factor is also important, and the environmental factor. It means that Jennifer and Karen, and usually they experience the same environment and same condition. So Jennifer and Karen eat nearly same food together, nearly same food, and at that they will show the 100% concordance. It means that it means that the question answer was the both genetic factor and environmental factor are important. And genetic factor should be same and environmental factor should be same. It means that they will show the 100% concordance in terms of their weight. Okay? And also in the dizygotic, even though the dizygotic shows the 100% concordance, it means that there are also both genetic and environmental factor are important. Okay. In this slide, we will see the adoption studies. The adoption study can distinguish the effect of the gene and the environmental on characteristics. Okay, the experiment is in this slide. The experiment is question is the body mass index BMI is influenced by genetic factor. Okay. So based on this experiment, we will see that the effect of the gene or the effect of environment on BMI. Okay. So this is the method is compare the body mass index of adopted children with those of their adoptive and biological parents. Okay. So this is the adoptive. This is the adoptive. This is the adoptive adopted children. Okay. Their children has their biological parents and also their adoptive parents. Okay. Okay. For example, this is the x-axis means show that the adoptee weight class, and this is the thin or the obese. The left is thin and left is obese. The children is thin and obese. Okay. For example, this is the BMI of the parents. The blue color is father and the red color is mother. Okay. If their biological parents, the father is BMI is lower, and they have a higher chance of the children will be thin. If their BMI of their biological father is higher, means that children will have the higher chance of the obese. Okay. How about their mother? Their mother is for example, if their mother BMI is the low, means that they have higher chance of their children is the thin, and also if their mother is the higher BMI, it means that they have higher chance of the obese, the weight. So this is their biological parents, but they also compare with the adoptive parents. So this is for example, their adopted children is one of them is thin and obese, and they also compare with their BMI of the mother and BMI of the father. So it means that the graph means that there is no consistent association between the weight of children and that of their adoptive parents. So, okay, based on this result, so overweight biological parents tend to have overweight children. However, in these cases, there is no consistent association between the weight of children and that of their adoptive parents. Based on this adoption study, the conclusion means that obesity is caused by genetic factor, not caused by environment. Okay. If their parents is the higher BMI, means that their children will have higher chance to get to be the obese. So based on adoption study, we can distinguish the effect of gene and environmental on the weight. Okay, in these cases, the obesity is caused by genetic factor, not environmental factor. Okay. In this slide, we will see the final subject. Genetic counseling provide information to those concerned about genetic disease and traits. So usually the genetic counseling is the new job So in America, there are lots of the people whose job is genetic counseling. So if you are interested in genetics, you had better take a job for genetic counseling. They have a higher salary in America. In Korea, we don't have enough people who have a who can provide genetic counseling. So if you studied genetics, it's very well. If you are interested in genetic counseling and study abroad, go to the America and study genetic counseling. And you can come back to Korea and you will have the higher salary or you will have the higher chance to get the job. Okay? So genetic counseling means that provide information related to hereditary condition. The table 3.6 summarizes the common reason for seeking genetic counseling. For example, the first reason is that a person knows of genetic disease in the family. For me, my father died from the colon cancer. Nowadays, I'm very worried about the colon cancer. So every year I receive the colonoscopy to check whether I have the colon cancer or not. So this is like me. Okay. The second reason is a couple has given birth to a child. Couple had children. The children have the genetic disease or birth defect or chromosomal abnormality. At that case, is this couple usually the seek genetic counseling, and after they did the counseling, and after that, genetic counselor is the calculated chance to for their second children. They don't have second children, but the genetic counselor is calculate the chance for their second child to have some disease or to have some birth defect or to have the chromosomal abnormality. And also the very common reason is that there are also several reasons, but the final reason is that husbands and wife are closely related. Usually, for example, consanguineous or where if they are the first cousin. So usually, in these cases, they seek genetic counseling. Okay, but genetic counseling is provide information to those concerned about genetic disease and the traits. Okay. So if you are interested in genetics, okay, you have to think about genetic counselor, okay? So next is the genetic testing provides information to those concerned about genetic disease and traits. Usually the people did ultrasonographic. In Korean, we call it as the 초음파. So based on this examination, we can estimate the age of the fetus, and also we can determine its sex, and also check the presence of the developmental disorder or any other problem. Okay, so nowadays people usually they receive the ultrasonography during the pregnancy. Okay. In this slide, we will see another genetic testing. We call it as aminocentesis. In Korean, we call it as 양수 검사. 양수를 뭐라고 하죠? 양수를 amniotic fluid라고 합니다. 양수는 여기 존재하고 있죠. This is the women who is pregnant. This is the baby. Between the baby and the women, there is the amniotic fluid is like this. Okay, so amniocentesis means the procedure for obtaining the fetal cell. So this is baby. Sometimes the cell is disconnected from the baby, and the cell is located in the inside of the amniotic fluid. So in these cases, Small amount of the amniotic fluid is withdrawn through the needle. Okay. After centrifuge, usually the sum of the fetal cell comes from baby is located. So usually after we separate fetal cell, we can culture and then we can do a lot of the analysis. So it means they collect small amount of the amniotic fluid which contain the fetal cell, and usually the amniocentesis can be performed early in the pregnancy, usually during the 16 or 20 weeks fetus. And usually we can do the chemical and the DNA and chromosomal analysis. So this is the amniocentesis analysis. Okay, 양수 검사. In this slide, we'll see another genetic testing method. We call it as chorionic villus sampling. So this is the women who is pregnant. So this is around 10 or 11 weeks fetus, baby like this. So here there is Korean. In Korean, we call it as 육모막. 육모막, there are several villus like this. 육모처럼 이렇게 막 나와 있는 거죠, 맞죠? So Korean is a membrane derived from the embryo. So this is membrane from the embryo. So nearly, not 100%, but nearly same genetic information as fetus. So usually they collect some small piece of the chorion from here, which contained, we can think that which maybe contained the fetal cell. Usually this can be performed in early pregnancy. So you will see here the 육모막 검사, Korean villus sampling is performed early in the 10 or 11 weeks fetus. So after that, we can do the testing, the chemical and the DNA and chromosomal analysis. So usually the amniocentesis is performed in the 16 or 12 and 26 weeks, 20 weeks, but CBS we can do in early status, can be performed early in pregnancy, usually in the 10 or 11 weeks fetus. Okay. Point 4 shows that example of genetic disease and disorder that can be detected through the genetic testing. For example, chromosomal abnormality, which they have or if they have the XXY or X0, Turner syndrome, Klinefelter syndrome. In this case is examination of karyotype from the cells obtained by amniocentesis or CBS. Also some kind of cystic fibrosis is also, and also DNA analysis of cell obtained by amniocentesis or CBS. Hemophilia is also, we can detect hemophilia by using the CBS or the amniocentesis. And also, for example, ultra osteogenesis imperfecta means that 골형성 부전증. So because they have some problem in the collagen defect, so usually their bone is very fragile. So they usually they can't stand up. So in these cases, usually the ultrasound or X-ray can detect whether the baby has the osteogenesis imperfecta or not. This is the example of the genetic testing can tell whether they have a chromosome abnormality or they have some disease like this. Okay. So final one is the final subject is the interpreting the genetic testing. So we have the genetic counselor, but even though genetic counselor is available, there is some complication of interpreting the genetic testing because some disease is caused by numbers of mutations. For example, cystic fibrosis. For example, in this cystic fibrosis we can detect the DNA analysis of cell obtained by the amniocentesis or CBS. Even though we can detect it, but more than 1,000 different mutations is located in at single locus of the gene, CFTR gene. So some diseases, and so even though we found some mutation, we don't know whether this mutation is induced cystic fibrosis or not. And also, some of them show the incomplete penetrance, and also some of genetic disease is affected by environmental factor. So we don't know whether this disease comes from the genetic factor or this disease comes from the environmental factor. That's why the interpreting genetic testing is very complicated and very difficult. But if you study genetic counseling, it will help people. It will help people to solve this kind of the problem. Okay. So nowadays, we will have lots of information. For example, after genome analysis, after we know all of the genome. Nowadays, our knowledge against the gene is increase and increase, and so some of the problem can be easily solved. So, but still have lots of problem in the genetic testing. Okay. Chapter 6. So we learned about pedigree analysis, and we also apply the pedigree, so whether they are X-linked, whether we can distinguish the autosomal recessive and autosomal dominant and X-linked dominant or X-linked recessive. And also we learned about the twin study and adoption can help to assess the importance of gene and environment. Finally, we also learned genetic counseling and some kind of genetic testing. Okay, we will finish today. We will finish chapter 6.